So we're continuing chapter 7. This will be relevant before we're done today. How the human egg is often deceived. All sperm around the egg cell. One of them's going, phone repair man, need to check your lines, ma'am. Another one says, insurance salesman, ma'am, mind if I stop inside? Package for you to sign for, ma'am. He said, none of you are old enough to know about land shark. Look it up on YouTube. We're going to talk about chromosome structure. Double helix, cross-connected at the A and the T and the C and the G. We've covered that before. Oops. I was going to tap the screen over there. If you stretch out your DNA, like took the DNA out of just one cell and stretched the molecule out, it would be about this long. And it's packed into a very tiny room inside each one of your cells. And that takes some packing. We talked about how chromatin, the material, is DNA and proteins. The proteins are spools. Is it spools and spool pilers and spool clusterers, it's in order to take and pack the DNA together and coil it up. And if the cell's going to divide, pack it really, really tightly. This is what a chromosome looks like when a cell is about to divide. It is as tightly packed as it ever is, because this is a copy connected to another copy, and they're going to be set up to separate, and then one of them's going to go into one of the new cells, one of them's going to go to the other new cell. While this is happening, this is so tightly packed you can't use it. You can't really get at the genes. If you need codes, you better have gotten them made before the cell starts to divide, before this gets really, really packed up tight. But under normal circumstances, an awful lot of your DNA is wrapped up fairly tightly. The stuff that you use is loose, but it runs through areas of the nucleus where some places it's easy to get at it, and some places it's not easy to get at it. We'll talk later about what's called location effect with a mutation that moves a piece of DNA from one place to another. These proteins are called histones. And the two together are chromatin. We covered that pretty recently. When you do make copies, I mentioned they got to stay connected for a while until you're ready to separate them out. Each one of these pictures, there's a little hash marked tight spot right there, sometimes close to the end, sometimes close to the end, sometimes more in the middle, sometimes right in the middle. That's the centromere. Is that has the specific purpose of when this is made into a copy, it's going to stay connected here. What you're going to do is pull these strands apart. I'll show this to you. Pull the strands apart, and with each strand, make a new copy of where the old strand used to be. Now, because what's on this side determines what's on that side, it doesn't exactly match, but if you have an A over here, you got to put a T across from it. That's what used to be across from it. If you have a C over here, you got to put a G across from it. That's what used to be across from it. You can make a copy of the other strand, but they do stay connected at the centromeres until you're ready to move them apart during cell division, which will be on the videos for the 4th exam. It's going to be in this chapter, but it's not. It's going to be a little bit too far in for the third exam. Up, maybe we'll put this up here. The very end of the chromosomes have a little cap on them, really tightly wound up chromatin. And that is called a telomere. You have a system set up in your cells that senses broken ends of DNA. Keep in mind, you got a very long strand of DNA, and on a molecular level, things sing and dance and bounce around and stuff, and so your DNA will occasionally just break. And so now you got two ends that used to be part of the same strand of DNA. You have proteins in your nucleus that go, uh-oh, broken end. Let's grab this. Got to be another broken end around here somewhere. Oh, there it is. Put it back together. We'll talk about that being a problem if you get multiple breaks at the same time, because typically it's only going to a single break from the shaking and dancing. Now keep in mind, your chromosomes have ends to them, and you really don't want your repair machinery to recognize that as a broken end and stick two chromosomes together tip to tip. That does occasionally happen as a mutation, and in fact it happened in our ancestors. I mentioned when I was talking about chromosome number that we have 46, and our relatives, the chimps, our close relatives, the chimps, have 48. Does that mean they have whole extra genes that we don't have? No, it means that at some point, since we shared an ancestor, and the ancestor had 48, how do we know that? Because one of our chromosomes, if you look at the genes on the chromosome, about half of them are genes that are on one of the chimp chromosomes, about half of them are genes that are on another of the chimp chromosomes, and you can actually see the remnants of two damaged telomeres in the middle. And at some point, there was a mutation where the two chromosomes were stuck together end to end, and eventually that became stabilized as our new number. So we wound up, one set went from being 24 to 23, right, 48 to 46. But you could tell by matching the genes up which chromosomes actually did that. And you can actually see the remnants of the telomeres there. So weird stuff happens. All sorts of weird mutations happen. But telomeres, now when I learned about this, back when mammoths roamed the earth, we never learned about telomeres. I mean, people knew that they were the ends of the chromosomes, but they didn't think there was anything terribly important going on there. Since then, this is what happened. People found ways to take human cells and culture them in a dish, get them to keep reproducing, and then you can test the chemistry of them. You can actually expose them to different toxins and expose them to different drugs and see what the cells do with them. We talked about that. It's a cell culture model. Not perfect because it isn't really integrated with the rest of the body. It also eventually becomes really good at living in a lab and not so good. It doesn't really resemble what it came from. But when people were first working this out, they found that sometimes you could get them to go for quite a while, and sometimes they would go for a while and then they would just die. And some bright person noticed that it depended on one aspect of the person that you got the culture of the original cells from. The older the person, the quicker the culture would die out. And people started to wonder whether we were looking at something that had to do with aging. It turns out it does. As you go through life, cells get damaged and need to be replaced. And in any neighborhood of cells, you have what are called stem cells. And so there's damage and a message goes to the stem cells, okay, we need some replacements over here. And the stem cells go, okay, let's make some cells and put them where they're supposed to go. It happens in some places very well, Your skin is doing it all the time. The linings of your intestines doing it all the time. Your liver's pretty good at it. Your brain is horrible at it. But so different parts of your body are pretty good at replacing damaged cells. But when you ask a cell to make more cells, it goes, yeah, okay, wait a second, let's check to see how set up we are for this. And the chemistry in the cell will check the, I'm losing the word. It goes over the DNA to make sure the DNA is in good shape. There's a word for that, and I can't get it out of my brain. If the DNA is not up to snuff, the cell won't divide and it won't make cells to replace the damaged cells, so you wind up with more damage. One of the things it checks is the telomeres. Why would the telomeres be a problem? When we talk about replication, making copies of the DNA, which I just realized probably should be here in the chapter, what happens is you've got two strands. Remember, you pull them apart. You're going to make a copy of the strand over here and a copy of the strand over here. That's done with a big molecule, a big enzyme that clips onto the DNA and rides along. It kind of unzips it. And then two new strands are attached as it goes. So it's riding down the DNA. It's riding down the DNA. Imagine I've got something like the size of, I don't know, a small dog going down my arm. And so on the backside of this, you've got copies being made. When that molecule gets to the end, it doesn't just slide off and make copies behind it. It gets to the end, imagine the dog on my arm, and just lets go. So when it does that, the two strands that are left separate out, but nothing makes a copy of the strands that used to be across from them. You literally have frayed ends on your DNA. Well, in cells that do a lot of dividing, like your skin cells and your lining cells, where you're always making new cells, telomerase is active. ASE ending, it's an enzyme, right? It comes in and it puts the two missing bits of strand on the tips. So it completes the telomeres, and the telomeres are now intact. For reasons nobody completely understands, not all of your stem cell populations do that, because as you get older, some of them just go, yeah, we got the code for that, we're not making it anymore. But we need it to fix the end of the telomeres. Yeah, well, sorry, it's on a timer. The timer says we're not making it anymore, and it stops making telomerase. So there are parts of your body that just are very bad at repairing damage as you get older because you're not making this anymore. You do make it in some places, but you don't make it anymore in places that you kind of need to repair damage. And that's one aspect of aging. So aging researchers are trying to find ways to reactivate the gene, right? Epigenetics, activation of genes, so that you do fix the ends of the chromosomes, and your cells go, yeah, okay, we'll divide and make more cells as replacement cells, and everything is wonderful. You repair damage. There's some evidence that that works pretty well. There's a couple of studies already that have managed to do that. However, because of the way that cancer works, cells divide and divide and divide and divide and divide. Cancer cells have some requirements in order to not die out as a cell population. They're damaged cells to start with. Most cancer cells die just on their own because they're messed up. But one of the ways they help keep themselves going is that they randomly turn on this gene or that gene that helps them. And it's evolution in a person, is that they're a little evolving group of cells that are just trying to be a great cancer, which, not great for us, good for that. It's very common for cancers to activate this gene again so that they can keep dividing and the cells don't go, wait a minute, we're too damaged to do this. And so it overcomes one of the impediments to the cancer cells just making more and more and more cells. So the cancer researchers want to be able to turn this sucker off so that they take away one of the adaptive advantages of a growing cancer. So they're saying to the aging people, you turn that back on, you're going to help the cancer cells that people get, especially as they get older. The aging people are going, if you turn this off, you're gonna cause a lot more aging damage, because even the cells that normally do this, they're not gonna be able to repair damage. They're not gonna be able to make more skin cells or more lining cells. So you've got, again, this upside downside of research into these particular areas. Okay, we'll throw one more thing in here that goes to the original cartoon that I put up there, because I can do this fairly quickly. One thing in humans that has to do with chromosomes, it doesn't with all animals, is gender. Now I gotta tell you, the terminology is changing, sorry. The terminology is changing, and I'm gonna give you the terminology the way I learned it. But it's in a state of flux right now, and this doesn't always mean exactly what I'm going to tell you. You'll find people that will argue with you on it, and they'll say, well, what you're calling gender is really sex. And the two words have kind of swapped, and I don't know if this is a stable change or it's gonna swap back. But the way I learned it is in organisms that reproduce sexually, and they have two partners doing it, right? You can do it with one organism where, you know, they can just re-swap out sets of chromosomes and make babies. And so that's sexual reproduction. The babies are genetically different than they are because they've mixed and matched the genes. But where there's two partners, what roles do they play during reproduction? That's what I learned was gender. And the classic gender roles, the one that's very, very common in animals and plants, is male-female. We're gonna talk about that in a minute. But that's not the only way. Some reproduction with two partners has no gender at all. They just swap nuclei, neither side's doing anything different. So that's considered to be genderless. There's no separate roles. In funguses, there's only two partners, but depending on the environment and the circumstances and the food and a bunch of stuff, there's a whole list of different roles that can be played by the two partners. There's one fungus I'm familiar with has 38 genders. Is that during reproduction, there's only two being played at any one time, but those two vary depending on the circumstances. So genders are sexual partner roles. And as I said, the most common one tends to be male and female. This all comes down to the cells that are made to get this accomplished, which is why a flower has male parts and female parts. You can have male and female trees. That's unusual. Trees are typically both male and female at the same time. But ginkgo trees are male or female. There are certain plants that have male plants and female plants. Some of them have male flowers and female flowers. And the things you think about when you think about the differences between male and female obviously don't apply to a plant. So throw out all that stuff. Throw out all the animal bits and pieces that you connect. This all comes down to the cells that are made. Males make sperm. Females make egg cells, technically called ova, but we're gonna just call them egg cells. And I'm gonna lead right off with the major difference. In the case of male and female, one of the two partners has specialized in giving offspring a head start, giving them food to help them develop, make more than one cell. It's going to start as one cell. Develop to the point where they can support themselves, right? Baby chicken and an egg, embryo and a seed, whatever you need. In humans, there's not a huge amount of food in a human egg cell because the embryo is going to develop for a while off of that food, but it's eventually going to settle on a surface and start to pull nutrients out of the surface and build a placenta there, because we work a little bit differently. If we were platypuses, which are also mammals, we would have pretty decent sized eggs, and the babies would develop from using the food in the eggs. But because you gotta hold a bunch of food. Egg cells, by necessity, have to be bigger, and sperm cells have to be smaller. And that works out to help the other role that the sperm now has. Now, you tend to think of sperm as having flagella and that they can swim. Some sperm do that, but not all of them. But one thing that sperm do have that's all in common of all sperm, from wherever they're made, they have to get to where the egg cells are. The egg cells are big and fairly stationary. They could be inside a female's body, in the base of a flower, released into the environment, but they're not going very far. And the sperm cells have a way of getting there. Yes, a tremendous number of them swim. Most of them swim with flagella. Some of them very rarely swim with cilia. Roundworm sperm actually crawl with pseudopods. But a lot of plant sperm travels a bunch of different ways because pollen is a casing with sperm in it. And all the different ways the pollen can spread from plant to plant is ways that the sperm have of getting to where the egg cells are. They land on the female part, they actually form a tunnel, the casing on the pollen forms a tunnel that the sperm can then migrate down to where the egg cells are. So it isn't always a swimmer, it has a way. Don't get trapped thinking about how this applies to people, and you're going to be wrong anyway. In any living thing that has male and female genders, egg cells are a big investment. A lot of food going in there. You can't make that many. Sperm have to be released kind of into the wild, and the odds of them actually getting to an egg cell are pretty lousy. So you better crank out a whole lot of them. So in any given living thing, these numbers are going to be way higher than these, even in things that produce many, 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 many egg cells. And things like parasites can produce hundreds of thousands of egg cells. They're making tens of millions of sperm. This is always a comparative number. Now, some people they think, humans, you only make one egg cell. Turns out you ovulate one egg cell, but every cycle actually develops anywhere from 4 to 7 typically. It has to do with the necessity of producing enough hormones to get the system working right. Is that it's kind of a holdover from when we used to produce litters, but we've kind of reduced that to only one offspring at a time. But our system, in evolution, you don't get what you need, you get what you get. And so we've compensated by extra egg cells that produce extra hormones for each cycle. So the idea of we only make one egg cell doesn't even work for people. So don't get that in your head. Lots more made in the sperm, fewer made in the egg cells. The process that we're going to talk about toward the end of this chapter, so after the exam, that makes these cells is called meiosis. Sperm and egg cells are haploid. They have one set of chromosomes. And they're going to make an offspring by adding 1 and 1, and the offspring's going to have 2, just like the mother and the father had, 2 sets of chromosomes. When you go to make these cells, the first thing that happens is you make a copy of all your DNA. I'll explain that when we get to meiosis, but instead of 2 sets of chromosomes, the very first thing you do is make 4 sets. And yet you got to get down to cells that only have 1 set each. Well, when you're making sperm, you've got to crank out bunches of them anyway, so you make 4 working sperm. But when you're making egg cells, you don't want to take all this food you're getting set up for your offspring and quarter it, divide it into fourths. So you keep all the food in 1 cell, that's going to be your final egg cell, but you got to get rid of the extra sets of chromosomes. And so you crank those out by producing polar bodies. Is that, so you wind up with one working egg cell and 3 polar bodies. And that's the list of differences between sperm and eggs. I'm gonna toss him down here. Yikes. One set of chromosomes is considered haploid. You occasionally see monoploid. They tried to change it to monoploid a bunch of years ago because that matches the Latin better, but it never caught on. And 2 sets of chromosomes is diploid. So 46 chromosomes is our diploid number. 23 chromosomes is what you get in a sperm or an egg cell. Later we'll talk about mutations that produce polyploid, extra sets of chromosomes, lots of sets of chromosomes. And that's very, very damaging in organisms that it's damaging in. Some organisms are like, yeah, okay, that's fine. And some organisms are like, nope, we're not surviving past a couple of cells now. Okay, we're gonna stop there. We're not quite done with this part of the chapter that's going into the 3rd exam.